Professor Connor, it's a pleasure to have you back on camera and chatting to us about emerging science and important areas in the field of macular degeneration. Firstly, just for those that may not be completely familiar with your lab, tell us about the scope of your work.
Thanks, David. It's great to be here. I really enjoy this meeting because we get to bring the experts of around the world here and really push all of our research forward. And so my lab is really interested in the role of the intersection of the immune system and neuronal degenerations of the retina that occur in macular degeneration, and how during different phases of these diseases the immune system acts. Can it be beneficial in certain cases? Should it be tamped down in others where it may propagate the disease? And so we're really looking at that intersection in our laboratory's work, and we're looking in two main areas. One is the complement system. This is like an innate immune system, which is highly conserved among species, most primitive on Earth, and how the complement system really works and functions in the CNS or in our brains and retina, which is part of our brain, and how it facilitates either degeneration or retinal tissue homeostasis on a normal basis. And the other side of the coin of the lab also looks at inflammation in the immune system, but the role of lipids and how bioactive lipids can modulate these inflammatory components within the eye.
So what are the most important areas of advance that you have seen over the last 2 years?
So, I mean, over the last couple of years, I think the role of lipids are really making a resurgence, and I think some of the work by Dr. Miller and Dr. Vavas looking at the role of statins in geographic atrophy or drusen formation are really one of the biggest future advances that I think need extra work, and I'm really happy to know them and know that work. Another big area I think is the role of the innate immune system in these diseases, and when it may kind of kick in, and does that innate immune or inflammatory response lead to increased disease pathology in individuals going forward. We see the innate immune system has been here with us for a long time as we've evolved, and before we had intervention therapies that we do now, such as anti-VEGF and other modalities, this was the first line of defense. But can it get out of hand, and can we see when it gets out of hand? Can we tamp that down? And I think that's another big area.
So turning now to the panel that you're going to be moderating, what is the subject matter, and what are the flow of questions likely to be?
So I'm lucky in some respects, is that we're the last panel of the session, which is emerging therapeutics in AMD. And so this will kind of incorporate, I think, everything that we learned throughout the entire meeting, not just in our session, which we have this really great panel of speakers, one looking at immunotherapy in CNV from Sarah Doyle from Ireland. We have Phoebe Lynn, who is looking at the gut microbiome and how that might change probably the inflammatory status of the eye. And we have Lindsay, who is looking at the genetics of AMD and how we might be able to pull out unique biomarkers. And so I think these three people have very disparate kind of research programs, but they all are kind of involving inflammation. I think that's a theme that we kind of see here. And I think by taking these individuals and then pulling in from everyone else that we've learned through the entire session, we'll be able to have a nice lively discussion to wrap things up, basically what we learned at the AMD biannual meeting this year. So I'm excited about that.
Excellent. When we look at immunotherapies, how do you see this field evolving? In other fields, in oncology, in rheumatology, biological agents that are highly targeted, monoclonal antibodies that are highly targeted, have been very, very successful.
Yes.
Do you see this as a path that AMD therapies might take in the future?
I actually absolutely do. But I think we're a little bit unique in the eye and in the brain in that we're somewhat immunoprivileged. You can't have normal inflammation. Like if we get a cut on our hand, you get this nice, heat response, you get this inflammatory cytokine release, you kill surrounding cells. You can't have that in the brain or the eye or in the retina because once you lose neurons, you can't recover from them. So the eye has our lab believes has developed these very tight regulation on inflammation, and we believe the complement system is part of that, and that allows it to specifically target injured or dying cells. And when that system gets awry in disease processes such as macular degeneration, that can lead to these confounding effects that we're seeing as AMD presents, drusen, geographic atrophy, subsequent neovascularization if the response is strong enough to break Brooks or the blood-brain barrier integrity. And so that's kind of where we see, like, what are these markers? When do they come up? How can we target them efficiently? And then with our genetics, those individuals working in the genetics field, well, they'll be able to tell us in timing and what markers might be most important. Because we have trouble, as you know, the animal models out there that, at least for the basic scientists, really we rely to model our animals on what clinicians see. And that's what I think is so great about this conference, is it brings clinicians and basic researchers together to help try and solve and talk about some of these problems that we see.
So in other words, you're trying to fill in the dots for a general theory of pathogenesis.
Exactly, yeah. Well, which probably, I mean, there's probably multiple pathways leading to similar phenotypes, but we feel that, at least in my lab, the immune system plays a major role in that, either in initiation or propagation of that disease.
So tell us about some of the next steps for your laboratory currently.
So right now, my laboratory is really looking at the role of the complement system in neurodegeneration, as well as vascular abnormalities such as choroidal neovascularization and diabetic retinopathy prematurity. And we're seeing this biphasic effect where we're finding that in these neovascular blooms, these proliferative abnormal leaky vessels that drive disease, they get targeted by the complement system for resolution. We feel that it's probably due to a lack of cell-cell contacts, and so here the complement system is beneficial. But in neurodegenerative diseases, when the complement system targets neurons that can't self-proliferate or renew, that becomes a degeneration over time, and we think that leads to geographic atrophy, and so we're interested in looking in that, in the role of the complement system in respects to different disease modalities.
What are, in your mind, the most effective modalities to therapeutically modulate the complement system?
So that's a really good question. That's something we struggle with a lot. We started with large-scale biologics such as antibodies for targeting the alternative complement system, which I think are out there in the clinic, but also small molecule inhibitors. There's pluses and minuses to both. Large antibody molecules targeting these proteins are usually somewhat restricted to the eye when we introduce them into the ocular environment. But then small molecules can either have a very potent effect when we introduce them to ocular environment, or possibly could be taken systemically. We worry about systemic administration because the complement system is really important for wound healing and our normal innate immunity processes. So we don't want to inhibit that for a long period of time. So we think targeted delivery is going to be important. Right now we have studies looking at small molecules as well as large-scale biologics. And I think you just have to see which fits best.
In terms of ocular delivery systems, what are the most promising?
That's interesting. I find Joe Cialino's contact lens, drug-eluting contact lenses, to be a really fascinating area. It helps, at least for small molecules, not maybe so much biologics unless it's on the ocular surface. But things that can penetrate in, this gives a long, sustained release of these compounds without the invasiveness that is seen with other ocular delivery devices, say, be it a needle or be it pumps that are currently out there and are being investigated. So
are you collaborating at that level?
So we first want to make sure we know what we're looking at. So we want to make sure that we have the right therapeutic targeting the right disease system before we take it into these complex biomaterials. And so we would love to hopefully get to that point someday, and hopefully someday soon. But I think the gold standard is not penetrating the ocular globe and still getting efficient drug delivery, and be that a lipid-based thing or a drug-eluting contact lens is yet to be seen.
For patients, they always want to know what are the potential implications for therapies for them, and how soon?
Yeah, I mean, that's That's the gold standard. And I think anyone, not only patients but investigators, they want to get their discoveries into the hands of patients as quickly as possible in the safest, most effective way. And if I were a patient that had these diseases, I would want to know that as well. And so it's a tricky thing because you don't want to lead individuals on. And so in my case, in my work, I think we're a little bit ways off from that, and I would never want to say, "Oh, tomorrow it's going to be tomorrow," because everyone wants it to be tomorrow, and so don't I. But the realistics for safety and efficacy, it's a big leap from animals to humans, and so I think many investigators are much further along than my group are in that field. But I think that's what's so important about bringing the clinicians and the basic scientists together to help streamline that process so we can get something to a patient's hands as quick as possible.
Professor, tell us a little bit about any other aspect that you'd like to highlight.
So as I kind of alluded to, I think we know AMD is this probably multi-genetic, causal disease, and it has, for clinicians and for scientists, it has kind of similar phenotypes at the end, and finding and elucidating what these different drivers of geographic atrophy, of dry AMD, of wet AMD are. As we learn more, they're not going to be one end-all or be-all. Kind of like, back in the 80s, everyone thought, well, cancer is this monolithic thing caused by one thing. Chances are AMD is basically the same thing. It's probably polygenic disease that's targeting various pathways that kind of have the end kind of stage phenotype in patients. And so what we need to do is start teasing apart what these different pathways are. Are there overlapping commonalities between these pathways, and how might we best target them efficiently and to help our patient populations type in patients. And so what we need to do is start teasing apart what these different pathways are. Are there overlapping commonalities between these pathways, and how might we best target them efficiently and to help our patient populations.
Well, thank you for your work. It's clearly going to be powering precision medicine for some time in this field. Well, thank you for your work. It's clearly going to be powering precision medicine for some time in this field.
Well, thank you.